Right, good morning, ladies and gentlemen, and good international collaborators. The first thing I have to do before you go anywhere else is to thank the organizing committee, more precisely Dr. Gita, for Dr. Gita himself brings the whole scene together in the NORCEL. But also thanks for this opportunity to present. Thanks for Martin Dominic, who is getting all the systems in place right at the beginning. And thanks also, do not forget, to Cathy McGrath, the redoubtable Cathy McGrath, for getting us organized. I shall press on immediately, even though I do seem to have got quite a lot of extra time. But I assure you, once I'm on this topic, it is very hard to hold me back. So this is, I call this microbiome studies. The basic principle being it covers a multitude of sins. And so what I'm really doing is I'm looking at the whole topic around the ages and then bringing it together. So let's have a look. The germ in the machine is my subtitle. This is an active replay, shall we say, of something I put in yellow to remind me. Arthur Koestler wrote the book in 1967. He called it The Ghost in the Machine. And he approached it from a philosophical and a psychological point of view that basically said, what is this thing we call consciousness? This is the hard problem. How on earth does a machine, a chemical set of operations, produce something that is conscious of itself? Of course, this question has recently arisen when an operator within a company said, I think that my artificial intelligence system is sentient. So I think I will leave that to Dr. Koestler and his advisors, and I will say that the germ in the machine is a slightly different topic. Of course, we know there are germs. There are germs all around. There are germs in the intestine. But the question is, what do they do? Are they just sitting there? Does the germ run us? Are we the germ? What is the story? So it's kind of semi-philosophical. Now, my fellow travelers on this operation, so Dr. Gita himself was temporarily left us, so you can tell him I praised him. Thanks to Dr. Gita. Thanks also and especially for a more recent assistant, and not just assistant, cooperator, Doctora Miriam Palacios Perez. He was also on her team, who was also in the Latin American pulpit. Now, this is indeed the 6th NORCEL conference, the first to be held in Scotland, in the historic university city of St. Andrews, which I can recommend to the entire world. And this is the first time I'm able to give a presentation on the microbiome, at least in front of a scientific audience. Now, when I met NORCEL, the emphasis was on HGT and LUCA, where HGT stands for horizontal gene transfer. And that the essence of this is Versa in 2002, but it's important to note that this was actually discovered before in the 1930s. But of course, they did not know the basis of the gene. So when genetics, the genetic mechanisms, the structure of DNA was discovered, for a while, this mysterious phenomenon was forgotten about temporarily. But then, of course, what happened? After the discovery of DNA, we're in the 1950s, the 1960s, we are in the middle of what I am terming the age of antibiotics. And antibiotic resistance became widespread, far faster than it should have. So this notion came again that bacteria, microbes, bacteria normally, can transfer their abilities, their genetic abilities, so-called horizontally, so that suddenly antibiotic resistance is very widespread in the environment. So this other word, LUCA. LUCA is the last universal common ancestor. So it is that theoretical entity which contains the genes that are now widespread through all the parts of life that we have been able to detect. So somewhere down the line, there was an entity or entities which contain these genes. So in essence, I will not speak much about Luca, but all I will really say, of course, is the essence of NORCEL is studying the emergence of life. How on earth life can arise from abiotic chemicals? And of course, this will be very useful for space exploration. The interesting point here, of course, everybody here, well, okay, the older ones, were born and brought up with Star Trek, to boldly go. And we know it is in our genetic inheritance to boldly go around the universe. So all these studies will eventually be useful. But there is, of course, a technical hitch. And I pull on a second main aim of NORCEL, that the Blue Earth project, we recognize that pollution, and I emphasize pollution, I'll come back to it, that the dual effects of pollution and climate change actually represent a threat to the ordered society. And if it is not brought under control, and if these problems are not brought under control, there will be no chance of space exploration for the future. So I think this is really critical. There's a limited amount we can do, but all we can do as our scientists, jump up and down and do our best to let people be aware that we need to make big changes. Now, another one, microbiome studies. You will be aware that between the emergence of life and the end of civilization as we know it, potentially, we have a gap of somewhere between 4 or 4,500 million years. And this really covers the process of evolution from microbes to man. And what brings me here today is the onset of something called non-communicable disease. And this varies from the trivial, when you get the sneeze in the middle of summer, to the really devastating. And at the moment, science, in my view, medical science, has not been taking this seriously. And this really represents another opportunity, because the threat to our society is as great as that of pollution and climate change. Now, there's an interesting point. So and I were discussing this, that really, of course, the microbiome project isn't really to do with the emergence of life, and it's not. But it's interesting that it is what I call a spin-off. Now, clearly, when President Kennedy decided to shoot for the moon, he was very successful, and they brought back a few rocks. And the other thing they realized was a lot of the technology was developed during this enterprise. Any enterprise that's worth its salt has spin-offs. And the microbiome is a spin-off. I don't know if this is the first spin-off, but in a sense, the Blue Earth Project is a spin-off, but not quite. Now, I like to put 3 points on each slide so I don't overwhelm the audience. Sadly, I'm not always successful. This is a case in point. But this line summarizes my entire talk. That the microbiome is the link between genes and the environment. Everybody knew when genes were first postulated that there would be a link to the environment, but nobody has really pursued this. The essence is all on the genes. So this is by accident, the first big break. So look, we can discover the relationship between genes and environment. And that, as such, it is part of the chemical informational universe. And that is why I'm giving this talk here today. Back to NORCEL, I think. Now, so let's say it again. Extreme claims requires very good evidence. So we really ought to look at the evidence. Now, to be fair, there's not that much evidence. I had to look very hard, but that's only because so far people have not been looking for it. So the key point really is the epidemiology, and we require reliable witnesses. We also require some context for the microbiome. So part 2 and part 3, which I will have time to present, plenty of time, is about the ecology and the evolutionary aspects of the microbiome. This, in effect, completes the picture, and that's very necessary. Moving on. So we have new disease or a new set of diseases. The who, the where, the when. These are important to know. When you've got these, maybe you can figure out the why. You will not get far in our writings before you discover Dennis Burkitt. Is he reliable? He was born in Northern Ireland, studied his surgery in the Edinburgh University Hospital. He was posted to Africa in the Second World War and went back to Africa afterwards, going around and looking at traditional African societies with his background and his knowledge. And in essence, in a paper published in 1973, which he published many papers, but I personally consider the one in 73 to be the most comprehensive. He pointed out that in traditional societies, there was no sign of non-communicable disease. He also, and this was a mistake, he pointed out that these traditional societies also had high volumes of dietary fiber because much of their nutrition came from roots. They tend to grow and store dietary fiber. So he then made the leap of adding 2 + 2 and making at least 5, that therefore all the disease in Western society were due to a lack of fiber. But it was wrong, and he knew it was wrong because in his own comprehensive studies, he pointed out that the Maasai are people who live in the steppe lands of Africa, not many roots. They tend to exist off animal products and grains in exactly the same way as we do in the West. Now, he sort of waved his arms, but the Maasai, the lack of disease in the Maasai was a really critical point, and he didn't follow this up. So he showed that when people brought up in a traditional society moved to Western civilizations, then they developed the disease. Whereas when people, but he never checked what happened when the Maasai themselves moved to such places. So I think sadly he's wrong. But again, I put the note that after the end of World War II, I call this the age of antibiotics. So I'm giving you a heavy hint here, ladies and gentlemen. But nevertheless, to find the beginnings of non-communicable disease in these Western civilizations, as opposed to the traditional ones, we have to look back earlier. Fortunately, there is such a person. Now, Dr. Bostock, he was born in Liverpool, in the United Kingdom, again studied his surgery in the University Hospital in Edinburgh, but he was a bit earlier, 1819. But he became a nephrologist, so he studied the distribution of urea between blood and urine. So yes, he was well regarded, so he's a good witness. And from his early days, he suffered from a very interesting phenomenon, that in the summer he would itch and sneeze. And he described this phenomenon, he called it catarrhus aestivus, and nobody had heard of it. Nobody had seen it. They didn't have a clue, but he was intrigued. So by 1828, he published another paper saying he had found 28 cases in Britain, and they were all from very wealthy people. Now, it is very important to note there were no antibiotics back in the 19th century. And I put in another note, smallpox vaccination, Edward Jenner, 1798, because people are tempted to believe that when things were reported from a long time ago, nobody really believes them. And I think that's completely wrong. Jenner was pushing, published in 1798, but he came up with hints from earlier than that. And so I think people have been inoculating one another for a long time. Do not underestimate the skills of people. So now my third point, and I stick to 3 points, but again, I'm cheating because I deal with 2 people. Now we're almost up to date, 1989. Up to date as far as I'm concerned, maybe not for the younger people. 1989, David Strachan produced a paper which has since been known as the hygiene hypothesis, which went viral. Nowadays, it keeps coming back reading about the hygiene hypothesis. And basically, he said that the immune system should be calibrated in childhood, or should be trained in childhood against external agents, which we are no longer seeing because we are too clean, too hygienic. That's what he said. Graham Rook took this up in my little note, Rook 2013. He said, no, we can't find, he gave up, his hands are up. We cannot find an external training agent for the immune system. Strachan was talking again about hay fever. So while Bostock spotted only 28 cases, now there are thousands, even in Britain, there are thousands of people. And the propensity for hay fever is increasing. And alongside that, we get asthma. And of course, we get deaths from immune system response with things like cheese and milk. This should not be happening. Alongside Strachan, we have Barker. So this is David Barker, later the University of Southampton, who in 1990 published papers which again could be classed as the fetal origins hypothesis. So that's gone not quite as viral as the hygiene hypothesis. The idea being that diseases such as obesity and schizophrenia had their origins in the womb. He said that the womb may be more important than the home, he said. Almond, in 2011, we published, and basically says the epidemiology is correct, but there is no sensible explanation for these findings. Now, let me focus now, because there are three things highlighted. So the immune system, I've said, covers from the trivial to the devastating and probably includes cancer. I might have the time to talk about cancer, see how it goes. This word obesity, I don't mean obesity, because obviously obesity itself is defined by some precise variation on weight and height, the BMI. But what I really mean is the propensity for the accumulation of energy within the body. And of course, that energy is stored as triglycerides. So it is just, so what I'm saying when I use the word obesity, because everybody recognizes it, is the propensity to accumulate that energy and accumulate it in the wrong places. In the viscera, into the bloodstream, you've got problems. Schizophrenia, that means split brain, and is representative of many of the mental health issues we now know of. But what's been happening very recently is that rather than just saying microbiome, we now say microbiota gut-brain axis. The microbes are talking to the gut talks to the brain. So the next slide gives us exactly that. Ladies and gentlemen, we have a gut, we have a brain, and we have an axis connecting them. That axis is partly neural, so we're talking about the vagal nerve, and partly there are chemicals pumped from the gut and from the brain, and they influence each other and they influence all the body. So this is how this axis works. So the actual publication that I'm referring to, the microbiota gut-brain axis in obesity. So I'm emphasizing microbes and I'm emphasizing obesity. And this came at a very opportune moment. For 2017 was when I was beginning to finalize these hypotheses. And so that one put the nail in it. When this paper came out, okay, I've sorted it. We look now. So we have, so again, this term obesity, this propensity to accumulate energy is always associated with the reduction in the microbial diversity. So the number of species operating in the gut is reduced. And that's associated with all these non-communicable diseases. We also know, and we are commonly told, that in order to be obese, in order to accumulate this, we have to eat more than we move. Now, okay, there's a lot of money involved in the supermarkets trying to sell you something. There's a lot of money involved in the gym trying to get you to move. But I have no reference for this one. This is what everybody has said, but evidence. Now, I also stress this other word, metabolism. And normally that can be considered to be basal metabolism, the energy you use when just sitting down. But maybe I'll come back to that. Okay, so I have a fact. They are few and far between, but here's a fact. Antibiotics are known to enlarge animals. The expression known, it is not broadcast, but certainly I gave this talk on an island in the Philippines, and they with the tourist industry, but they also grow their own food, and they buy the antibiotics, they grind them up, they mix it with the feed, and their animals get larger. In the West, our animals don't get obese, they get slaughtered. But pets animals, they certainly put weight on. I have three references. Davison in '83 talks about the enlargement of chickens. Cromwell in 2002, the enlargement of pigs. Radar 2013, the enlargement of fish. These are really talking about the enlargement of vertebrate entities. Of course, we don't really farm anything else at the moment. Insects are a common. The I'm guessing the bottom line, presumably antibiotics, if they do not reduce the diversity of bugs, they certainly may affect what these bugs do inside the gut. They make them ill. And so there's something there. Now, when we move across to people, life gets more tedious. So one statement that I can make is that calorie intake has fallen over 80 years. This is calm. I actually brought some papers, but they're not easy to pass around because you're too well spread. I brought a couple of papers. You can look at it if you wish. But basically, the reference by Calm in 2005, and what he said was that basically summarized work done earlier in the Second World War. So my second reference down, Ansel Keys, in the middle of the Second World War. America was winning this war, but it knew that when it won, it was going to be faced with millions and millions of starving people. So Keys did experiments. It's called the Minnesota experiment, the Minnesota starvation experiment. He took volunteers, conscientious objectors, and found out how much they normally eat, and then put them on a reduced calorie diet for 3 months. And these people were devastated by this reduction. But the numbers are interesting. As given here, 3,200 kilocalories per day. Nowadays, the recommendation, which I think many people follow, is for 2,000 kilocalories per day. Now, the interesting thing, talking about fiber, because I listened to Dennis Burkitt. And in the 70s, 80s, 90s, I ate high fiber, as you're recommended to do. Ladies and gentlemen, I grew fat on high dietary fiber. What I did then is I just simply cut down everything, ate far less. So nowadays I probably eat 1,800, 1,600 kilocalories per day, and I am not starving. So things have changed. I have this data for 3,200. People might have difficulty believing it, but I have backup information from 1920 from Britain, which says that active women folk, because it was aimed at women, should be absorbing 3,100 kilocalories per day. Sorry, but she also pointed out, study done by a woman, that inactive women should be eating 2,250. This is a very precise number. They knew all about calories since the turn of the previous century. So there's no kidding here. I can also state that the level of exercise was unchanged because the Westerterp and Speakman did a properly physics experiment with double-labeled water. And across the period 1980 to 2008, there's really no difference in energy use, even though there was more use of cars and more use of machinery. We stayed more or less the same, but we grew fatter. We grew fatter over that time. The weight increased. So what's going on? Well, to be honest, at this point, I can give you a very interesting hint. The hint is I will give you a bucket, and in the bucket is labeled Burkitt, 1973. Because, ladies and gentlemen, when Burkitt studied the people's untraditional societies, most of them living on high fiber diets, he pointed out that the fecal output from these people was 3 times as great as you find in modern civilization. And that I replicated that data from information in the sewage industry, which said that in lower income countries, the output is greater, fecal output is greater than it is in high input. And of course, they all say it's fiber. I say it's not fiber, ladies and gentlemen, it's not fiber. But the hint was there. So in other words, it is really the difference nobody measures is what's going on in the fecal output. So we published this, Smith and Jeter, we published this in 2021. Again, thanks to Sohan for assisting all these processes. We've done quite a few publications now. Beginning to get almost respected in the field, if only people would read the damn things. Suffice to say, we published this in 2021, and we call this microbiome gut dissociation. And we referred to the reason for obesity. And we said, look, the reduction in fecal energy excretion. And we can trace all these back, but we can see the diversity of the bacteria that are within feces. We can look at diversity But nobody really looks at the quantity, the energy, and they should. Now, this is to do with the microbes and the development of obesity. And of course, I've got plenty of time. The next question is, what does it do to the brain? So again, we have the gut, we have the brain, and we have the axis. And the publication is Barker's publication I mentioned before. So this is the fetal and infant origins of adult disease. But I stress again, the infant origins, not just the fetal. So a reminder that studying the epidemiology, he said that schizophrenia, which we call split brain, occurs in the child right from the beginning. So down this axis, so put some nice expressions. One is this word interoception. Perception, we understand. Interoception is the knowledge of what's going on inside your own body. So it's perception of what's going on inside, because it's largely subconscious. And this is associated with intuition, which we know of as gut feelings. The expression is commonly used, but there's never really investigated. Even medical guys say, what's your gut feeling? Well, what does this gut feel? What does it mean? So I've put a little note to myself that says Savant, Miller, 1999. What I'm referring to is what's called Savant syndrome, which people who are quite seriously disabled, nevertheless have huge reservoirs of brain power. They can do music, for example, mathematics. Okay, so our view on this has been that we say that microbiome gut dissociation occurs in the neonates. So as soon as the baby is born, the microbes have to start talking to the gut has to start talking to the brain. The microbes aren't working, it doesn't talk to the gut, the dissociation that we refer to, but then what happens to the brain? So as the neonate develops, ladies and gentlemen, it's looking for connections. It only emphasizes those parts in which it gets connections. If the gut is not talking to the brain, that element is not emphasized. And so in preparation, we have another publication, probably the last current series, perhaps, is, and we refer to autism. And autism is the great unknown. Nobody really knows what causes it. Now, an aside, speaking of talking about, is that, of course, 20 years ago, somebody did think they knew what was the cause of autism, and they published it as being connected to the MMR vaccine. And that clearly was not true, and he was jumped on. So nothing to do with the MMR vaccine. But then it became, how should we say, went the wrong way, broke bad. And now every time there is a populist, you can be sure, populist politician, you can be sure that somewhere in the train, this individual will be represented. And he carries quite a good selection of fake news with him. So the understanding the cause of autism, you couldn't really get more important than that. So this has a chance to be a huge thing. Now let's see what happens in the individual and the person themselves. So in the womb, it is clear, we develop according to our parental genetic inheritance. Now there have been reports that there are bacteria present in the womb, but at the moment I'm not inclined to pay much attention to that at the moment. I could be completely wrong. Nevertheless, when the baby emerges from the womb, of necessity, there is fecal contamination. And I'll put contamination in brackets, because this contamination is part of the process. And as that contamination goes from the mother into the intestines of the child, it develops, and it ought, at that point, to be talking to the gut wall. So there are two inheritances, ladies and gentlemen. There is the parental genetic inheritance, but there's also the maternal microbial inheritance. And it is the interplay between these two that is under discussion. I would like to say also that in the maternal inheritance, it carries the seeds of the immune system to calibrate the immune system. That's my understanding. That's the best explanation. So on this slide, we deal with both Barker, which I mentioned previously, and Strachan, which I mentioned previously. this again, this principle of this is Jeter and Smith, 2019. I'll come back to that. But now also, having talked about Strachan, we can go back to John Bostock and his paper in 1828, in which he mentioned that it was associated with wealth. And now we have a hint, and this puzzled me for quite a long time. How can he be doing this when there's no antibiotics? Oh, of course, I thought to myself when I realized that in those days, people were using cosmetics that carried toxic elements. So white lead would be used for a blusher, white lead carbonate, red lead oxide for the lips. Black lead could be used for a beauty spot, and also, of course, used back in Egyptian society. So wherever there's been large, complicated societies, they've used these things, even back in the Neolithic and probably in the Paleolithic. You can see little pots, and then when you look at the pots, you can see these toxic cosmetics. Now, down at the bottom, I have another note, a guy called Paracelsus, which is easier off the tongue than Theophrastus Bombastus von Hohenheim. He was known as Paracelsus for some reason, and he is, and he pointed out that the poison is in the dose. They knew back in the 1530s and before that really below a certain dose there is no toxicity. So when these people were putting cosmetic lead salts, they knew they weren't going to get poisoned if they kept it down. But nobody was measuring the microbiome, and nobody was measuring the effects of a bad microbiome in the child, especially since that child had to develop into an adult for these changes to become obvious. There's an uncanny resemblance here to the disaster of the thalidomide, in which women were treated with this agent, but you could see immediately the results on the child, and you didn't have to wait till they were an adult. And even then it was hard enough to associate these deformities with thalidomide itself. So what we're actually saying is that when you have these lead salts or antibiotics or delivered by cesarean section, what we're getting is maternal microbial dysbiosis. And this dysbiosis is a word that is commonly used. What does it mean? Well, this is my next overhead, which we are talking. So this is another article by Harold Brousseau, and in 2019, and he said problems with the concept of this dysbiosis, and he was talking with reference to the probiotic industry, probiotic bacteria. And what he said, and I brought a copy of it, but again, I haven't passed it around, maybe you can look later if you're interested. I call it the angry article. He said, look, there are no causal relationships. There is no sound ecological or evolutionary reasoning. His article was quite interesting. It was quite an angry article. And in fact, what he's really saying is there's no such thing as hot postulates for these bacteria. So if you look again at the industry requirements, and this is, please note, this is not just industry, the industry alongside the regulator. And what we're looking for is a single patentable bacterial species. This idea of single entity, patentable, bacteria, specified. It's beginning to believe that bacteria are behaving like chemicals, and bacteria are not chemicals. You cannot use in the same regulatory framework. No, sorry, it shouldn't be done. And in spite of all this I've said, it shouldn't work. This is a very successful industry. So people like their probiotics, they buy them, the money comes in, they then pay for more research along the same lines. And then I think it was Brousseau's last start as he retired. So let's have another look. What does the microbiome actually need? It needs a high diversity. So it's one entity going to provide a high diversity? The answer is it's not. So the other aspect to our publication and preparation at the moment is that we're going to call probiotics a placebo effect. Now, it's long been known that the placebo effect is very powerful. It has long been said that if only we could pop the placebo effect into a pill. And it sounds to me like that's exactly what the industry has been doing. Another 10 minutes. No, that's perfect. Perfect. The so this again was Jeter and Smith 2019, a new way to view the human intestinal microbiome. But now what we're saying is that dysbiosis is not because of specific probiotic entities. This is because of the function. What does the microbiome supposed to do? So at this point, I will move across. We bring back horizontal gene transfer. Versa 2002. We're looking for an ecological rationale for the functioning microbiome. What's it supposed to do? And here we have the viral shunt mechanism. Been reported to Wilhelm in 1999. When you eat food, you take in nutrients. That's obvious. We also take in microbes. And those microbes come alongside microphage viruses and with mobile genetic elements. And I put a little note, these are circular DNA. The DNA you find inside microbes is prone to be circular, joined, connected. The picture of the virus is from electron microscope image. Tidied up, but that's where it comes from. And you actually, in the same electron microscope, you see lots and lots of circles of DNA. When you cook and digest, of course, the nutrients for growth, movement, and metabolism. But in the microbe, in the intestine, we get these phage viruses, we get these circular elements. I've drawn these little images with some stretches of DNA, but really these stretches are circular. And it occurs to me that these circles are circular because they can be more readily transferred from one bacterium to another, from one microbe to another. So in other words, it's almost as if the system is designed to be destroyed. These phage viruses probably, they don't destroy the bacteria. I think they disassemble them. And then they can be reassembled. And that reassembly constitutes this viral shunt mechanism. So really, it's a way in which a microbiome, a community trapped inside the guts, can change its composition over days by disassembly, reassembly. So I think this is built into evolution. We published this, Smith, Jeter, Palacio, Perez, in earlier this year, 2022. And our summary is environmental information is passed on by the maternal microbial inheritance. This information comes down from the outside via the food into the intestine, but there is no defense against heavy metal toxins. And a nice English phrase. The heavy metals put the spanner in the works. They stop it, they block it up. Why? Let us look at the origin of the microbiome. Evolutionary rationale. Also its inability to deal with heavy metal ions. Let's look. I'm talking about the Precambrian. The Cambrian was a period, I think the period about 500 million years ago, when we can see fossil evidence of all the entities we see today. So the Precambrian is all that time before 500 million, which is still a good healthy chunk of time. And this is the era of Darwin and nowadays Dawkins. So this is the survival of the fittest from Darwin. Neo-Darwinism says the survival of the fittest gene. And the expression then, though nature red in tooth and claw, that comes from a poem, but I think it summarizes the times. Nature may be brutal. Mankind, of course, is not brutal at all, as we know well. So if we put into this idea, the zeitgeist, the spirit of the times, competition, then throw Lynn Margulis and her idea that symbiosis is really the core of evolution. It's evolution by cooperation. At this point, fur and feathers flew. Big battles between the proponents of cooperation, symbiosis, and battle. So this is my summary, our summary, that in the microbial community, we have eukaryote, originally we had entities called prokaryotes. And these are the simpler ones, including bacteria. Margulis' idea was that the combination of prokaryotes leads to a eukaryote, such as amoeba. So this is what Margulis was trying to say. We're getting the more sophisticated ones within the microbiome. Evolution gives rise to multicellular entities, but the communities remain. So far, this is unpublished, but I think it's logical. So the evolution produces the teeth and the claws, as referred to in the poem. The microbiome is the immune system. We've published that notion. The microbiome contributes to the immune system many times by sentinel cells, and nobody has complained. A summary of the viral shunt and the context, viral shunt mechanism, out of the Precambrian, mobile genetic elements, phage viruses, prokaryotes, unicellular eukaryotes, and also the multicellular entities, including representation from all the above. And that's what's forgotten about nowadays. Plants and fungi, they grow in certain areas. These areas can be contaminated. Plants and fungi can't move. So they develop methods of sequestration, which we can use for bioremediation. Plants grow, they sequester the toxic chemicals, and they're taken away. Mobile things, invertebrates, vertebrates, they had no such mechanism, no sequestration. The evolution occurred outside. What we've done is we've taken pollutants and spread them around the world. Consequences of the disablement of microbiome. This is the last slide. Damage in the adult, the newborn, and what we need to do in society. In the adult, damage is done in the neonate. We can only control careful eating. And important, most important, nutrition in childhood. And Sandercock, a reference, talking about what's happening in children. The newborn, in principle, we can prevent disease in the future. We identify key microbes, probably the eukaryotes, and these probably constitute a missing link between the microbe and the people. LaForest Lapark, 2018. In society, we have to sort out pollution. This is back to the Blue Earth Project. We have to develop, we can develop phage therapy and maybe intravenous antibiotics that can't in themselves directly affect the microbiome. We need reorganization. And finally, thanks again to Dr. Jeter, NorCell Committee, for giving this opportunity to talk to you. And I hope that's adequate. I don't know if there's going to be time for questions. That's over to the committee.